Let's start. What I wish to discuss now is the epidemic from the point of view of the virus. There is, in all the things we discussed until now, we took an anthropocentric view, and I think this may mislead us in terms of understanding what happens in the cell and how the virus will evolve. In particular, there is something that people often don't understand, is that if I can speak of an aim for a virus, the aim of the virus is to make other copies of viruses, not to kill its hosts. Okay, so that is an important point, because in general, the virus won't evolve towards a more dangerous form. Okay, so I think this may be important to know. Now, let's think about how do we see life. Typically, the general property of life in terms of its physical chemistry, it's growing. It's always growing, and all chemical forces in living systems are poised in such a way that the cells will grow. So I will give you an example of a physical system which we generally do not take into account, just to show you that all physical things may happen, and then we will go to metabolism and chemistry. So I used an example that was suggested by Victor, in fact, some time ago, which is what could be the influence of gravity on living systems. And the example was motivated by the architect Gaudi, because the way he used gravity, and if I understand correctly, the Sagrada Familia is still not completed, okay? And this has been used also in St. Louis, in Missouri, as a gate, which is an arch also based on understanding gravity. So Victor proposed that we had Gaudi's principle to see how, when you perturb a living organisms, you let it evolve, and it will evolve following a physical-chemical principle, and this was illustrated by gravity. And well, you may say, oh, gravity is not a good example. Well, there are experiments where gravity is used, and it has been shown, and this is actually surprising because it's true even for bacteria, that gravity influences life. But of course, this is something very limited, and this is not what we will discuss now. In fact, what is providing most of the physical constraints in cells are chemical reactions, which are driven by the standard thermodynamic constraints that we know. There's one component here which is important and which is usually not very well understood. It's the entropy compound, which means it has nothing to do directly with order or disorder. In fact, the entropy tendency of things is to occupy as many energy and space states as possible. And I think this is something very important. It can lead to order, for example, by shaping macromolecules. The increase in water entropy makes it that you make a double helix of DNA in water, not in dimethylformamide, for example. So many biological processes are entropy driven. And this is something important to have in mind. Now, how are things linked to enthalpy and growth? And this is linked to the way chemistry is embedded in a cell, and in the fact that when you change the cell's volume, there is a lot of properties of the cell which are changing its chemical reactions. And we will try to elaborate on that. In fact, some of you may remember that I discussed that with Pablo in particular, because I'm still looking for people trying to work on the conjecture that there is something what I call non-homothetic growth. You will see what it is, which makes that cell growth has very important consequences. Typically, there are two types of life states: growing life, where the organism is reproduced while its program is replicated. And remember that reproduction and replication are not the same thing, despite the fact that they are often taken as this, using the same word. And stationary phase, which is survival, which is our state, in fact, which is also the state of infected cells. And there you have maintenance of components, maintenance of membrane potential, maintenance of energy supply. And the thing is that the chemistry of life must accommodate both things. And this is really surprising. In fact, this makes things very difficult. Metabolism must accommodate growth and stationary phase. And because there are no change, there are changes in the reaction, the level of reaction, so on, but the components are the same. And in fact, growth may affect form. And this is known, this has been emphasized by Darcy, Thompson, Waddington, and many people. And this must also play a role at the molecular level. And we will go into that now. So as you see, this is typically the, I would say, Greco-Latin way of things, presenting from very general to become progressively more detailed. So we have to think of the metabolism in 3 dimensions. Usually we tend to write things on paper like one molecule and an arrow giving a couple of other molecules and so on. We forget that all these things happened in a particular volume. And when the volume increases, this is very surprising because the increase of the volume, especially if the volume is a sphere, will imply that if you want to keep everything equal, then you have a lot of things to be done in parallel so that you can fill in the same volume with the same components, okay? And this will be very different from what happened, for example, at the surface of the cell, because for the cell, the surface needs much less components than the interior of the cell. Typically, this is something that viruses will have to see because, of course, virus infect cells which are not growing normally. But because cells have been devised to work as a growth system, in fact, they had to accommodate growth of the cytoplasm, which is in 3 dimensions, growth of the membrane, which is in 2 dimensions, and growth of the genome, which is in 1 dimension. And putting all these things together is really not straightforward. And actually, this general property is known in economy, and it's named under the name of non-homothetic growth, because it means that you have parameters which grow with a very fast speed of large increase, whereas other things cannot follow or must not follow. And so in order to keep all things equal at the end of the day. So typically, some people were interested in that for cells and wondered about cell shapes. And there are a lot of theories which try to explain why cells are generally not spheres. And they use different ways to explain that. Typically, Rayleigh principle of energy dissipation or things like that to explain how a cell would grow. Curiously, to my knowledge at least, and if some of you know more, I would be very much interested to know how it is. Most people don't consider at all metabolism in this picture, which is strange because, of course, if the cell is growing, if the envelope or the membrane of the cell is growing, it has to use metabolites. And these metabolites have to be synthesized, and this must have very specific constraints. There has been recently a model, well, 5 years ago, a model which was quite interesting, explaining why bacteria are in general bacilli rather than spheres. And again, it did not explain why metabolism or how metabolism would be involved there. Of course, implicitly, you can see that becoming a bacillus makes that rather than growing at the third power, you will grow rather at the linear, in a more linear way. This is a way to accommodate non-homothetic growth, okay? So in any event, at some point, we need to see how metabolism would get in. So now just a small parenthesis, which makes that I think many people did not try to think in terms of trying to find out a solution for non-homothetic growth. It is that when you look at living organisms, you find that most often they are far from perfect. That is, they adapt by recruiting what people name kludges, which or patches in computer programs, which are just small things which are added to something which is conceptually well designed and consistent, but cannot work well unless you add this kludge. And I think this explains why this kind of anecdotes that you find everywhere in biology, which explain why there are millions of papers which are published, because each one describes one tiny kludge, whereas they are not interested in the general concept or something like that. So now we have a general concept as a question, which is how does cells resolve non-homothetic growth? So if we were faced with kludges, that is one solution per organism, well, this would be hopeless. And I think this may explain why people are not really interested in finding out how all this works. But we also could be lucky. In fact, we could find that there was one of very few solutions of this general problem. And then this would be really very interesting, because this would be more or less universal. And this is more or less what I think we find here when we study growth and the way cells can harmonize the growth of the cell, the growth of the chromosome, and the growth of the membrane. Okay, so typically membranes, as I already told you, membranes have a shape which usually differs from that of a sphere. Well, and there are explanations for that. In contrast, DNA tends to increase in size, and in fact, if you look very carefully, you find that what is within cells, in DNA, the number of genes and so on, is fairly large, and you have the impression that many genes are more or less useless and sometimes degenerate or something like that, as if we had too much DNA. So this suggests that there is a pressure for DNA to grow, and this would adapt very well to the idea of non-homothetic growth, because you have metabolism in 3 dimensions, DNA is in 1 dimension, so you make far too many precursors, so it tends to increase in size. And we need to understand how this goes. And as you will see, for DNA, we already find something very specific involving cytosine, and this will be a hint to go further and see what happens in general. So typically, cells are constructed from a limited set of building blocks, okay? Amino acids, nucleotides, lipids, and sugars. Typically, one might hope that in this limited set, especially amino acids and nucleotides, there could be a choice that is used to adjust the growth of the different parts of the cell, namely the cytoplasm, the membranes, and the chromosome. Okay, so there should be some places where you find some kind of a rheostat, that you can adjust with the growth rate, and even when the cell just have to turn over because they are in stationary state. Of course, just a rheostat is okay, but it needs also something else. As usual, you may remember that I like to see things starting from functional analysis, that is starting from function rather than from structure, and get to find out what structures have been recruited for a particular function. So typically, a rheostat would not be enough because you need some kind of inertia, some kind of flywheel. And typically, it's important to see when you identify some component, whether it can be also associated to something which can act as the flywheel, that is a kind of store of matter, energy, and so on, so that you can use it when you need it and accumulate it when you don't need it. So now DNA is linear. There is no problem with that. And energy in the cell is available through the nucleoside triphosphates. And the driving force for construction of DNA, and in fact for construction of everything in the cell, is pyrophosphate hydrolysis. If you go to the KEGS database of reactions, you find that more than 500 reactions are driven by pyrophosphate. The reason is that once you make pyrophosphate, it is hydrolyzed, and hydrolysis is completely irreversible, not because water is in excess, which is already driving forward things, but especially because the way the reaction is poised needs not only water, but also metals, which need to be associated together in a very difficult organization within pyrophosphate hydrolase, which pyrophosphatase, which makes the reaction totally irreversible. And so typically it is availability of nucleoside triphosphates which acts at the major metabolic force for build up things. And what is built up are nucleic acids. And now I spoke about flywheels. Because they are macromolecules rich in phosphate, typically they can, by themselves, act as a flywheel, especially RNA, which is both synthesized and degraded continuously. So the enigma of DNA synthesis is here, and it's really surprising, and it's a bit strange that it has not been more emphasized. To make deoxyribonucleotides, most cells use ribonucleoside diphosphates, not triphosphates. And this is really unusual because in general, you will use, of course, the triphosphate. And especially because of what I just said before, you need to drive forward the reaction. You need to make pyrophosphates, and hydrolyze it, which cannot be done with diphosphates. So the interesting thing here is that to make DNA, in fact, this means that you have NDPs, the diphosphates available, which are at a very low concentration as compared to the general triphosphates, about 1%. So if we think in terms of the metabolic pressure on DNA construction, that is, once again, from a 3D metabolism to 1D metabolism, you already gained a factor of 100 in the pressure. So that is, the pressure on DNA synthesis is much less than it would be if it used triphosphates. I won't discuss now, unless you have questions about that later on, how this could be modulated at some point by using triphosphates also. So the problem, this opens up a very dangerous problem, which is the following. Because DNA uses, in order to make dinucleotides, not deoxyribonucleotides, you use dinucleotides, the position of UDP would be central and very dangerous because you do not want to have UDP in DNA, okay? Because of course, nucleoside diphosphatoreductase can immediately reduce it to DUDP. Then with the nucleotide kinase, you go to DUTP, and this would go to DNA, and you don't want that. So this means that evolution had to create another annex pathway, which was also driven by pyrophosphate hydrolysis, by the way, which is to hydrolyze DUTP specifically into DUMP in order to make thymidine and DTTP getting into DNA. So this is universal, and this is a bit surprising. And it shows that we have a problem with the management of UDP. So if what I said was important, then you expect that there should be arrangements in the cell to control UDP very tightly. As a matter of fact, if you look into uridylate kinase, at least in bacteria, this has been shown in E. coli, Bacillus subtilis, and Mycobacterium tuberculosis. And this is really strange. The gene which codes for uridylate kinase is located in an operon which codes for translation, a bit strange. And also the protein itself makes tubes which are located more or less around the surface of the cells. So it's highly compartmentalized, allowing the cell to control the UDP concentration in a nice way. Now, there's a second thing which shows that this is linked to DNA. It is that if you use a MUC-B mutant, that is a mutant which interferes with segregation of chromosome in E. coli, and you look for suppressors of MUC-B, you find that the suppressors are located in PRH, the gene for uridylate kinase. So there is a link, at least phenomenologically, phenotypically, between chromosome segregation defect and synthesis of U, which is an interesting thing. And this goes further. This goes here. There is a second suppressor, which is a suppressor of PRH, which is a suppressor. But I'm sorry, because it's really a story for geneticists who like to make mutants and mutants of mutants and so on, and find how they are interconnected. But what happened that people discovered the suppressor of PRH, which is a suppressor of MUC-B, and named it MSSA. And later on, it was discovered that this MSSA was the gene coding for citidylate kinase. This is something really surprising. And it was always in operon. You can see it in Bacillus subtilis and Haemophilus or similar Rhizobium melioti. It's in an operon with a ribosomal protein, RPSA, which codes for protein S1 of the ribosome. There is no S1 in Bacillus subtilis. And in fact, as you will see, the function of this protein is not simply to recognize the start of a messenger RNA, but in fact to present the start of a messenger RNA to a degradation system. In fact, RPSA codes for protein which contains a motif named S1, and it's present in many other RNA binding proteins of bacteria. And it is associated in bacteriophage T4 with a specific RNAs which cuts mRNA at GA-GG motifs. So typically something you recognize at the Schneider-Algarno as the ribosome binding site in messenger RNA. S1 is also a subunit of bacteriophage Q-beta replicase. So as you can see, these things will always link citylate kinase, well, nothing to do with cytosine at this time, with turnover of RNA. And typically, to make a long story short, the function of this operon is to make CDP for DNA synthesis. As I told you, we have a problem to make DNA. And in particular, I will emphasize that now, you will see that the very fact that the synthesis of C does not go through CDP implies that you need to have degradation system to gate it. So typically, here we have the following. The degradosome, for example, in B. certilis, E. coli, tuberculosis, and so on, contains PNPase, which is an enzyme which uses phosphate to have phosphorylysis of RNA, giving directly the NDPs. You have poly-A polymerase, RNase E, S1, the protein S1, etc. And this allows you to make CDP for de novo DNA synthesis. And also, by the way, GDP to recycle GTP for carbohydrate secretion. And it's interesting because the enolase, when it is bound to the membrane, uses GDP and not ADP with phosphoenol pyruvate to make GTP. Remarkably, in eukaryotes, you find the so-called exosome complex, which is located usually in the nucleus, which has very similar properties. So all these things meant to make CDP, I would say, are conserved throughout evolution. So how does cytosine enter DNA? The question is the following. In order to make cytosine derivatives, you start from UMP, as described here. You make UDP, then UTP. You saw that this was a problem because you had the diphosphate, ribonucleotide diphosphate reductase worked on diphosphase, nucleoside diphosphate reductase. And you make CTP and you do not make CDP. So there is no way to make CDP. So normally, if I would say metabolic models were something really useful, they should have found that with what we know about metabolism, generally, it would be impossible to make DNA unless you enter macromolecules in the picture. And this is something really important. In order to make DNA, you need to make RNA turnover, as described here by the red arrows. You can see that there are two ways. One, which is simply by hydrolysis, and this will lead to citidine monophosphate, which citylate kinase, which will citidylate kinase, which makes CDP, okay, which goes then to DNA. And the other more economical way, which uses polynucleotide phosphorylase by phosphorylysis, which makes directly CDP. So in order to go with DNA, to make DNA, you need to go through a very specific way of using cytosine nucleotides. And here you can see that if you want to adjust all these parameters, you need to control the enzyme, which is UTP kinase, UTP deamidase, which is CTP synthetase, coded by PRG, which acts as a rheostat. Exactly, it's coupled to nitrogen sources and to energy consumption. So you have something which very finely can tune the way you enter into DNA. There is a second rheostat here, and I thought about that after discussion with Victor, in fact. It is the whole setup which goes through pyrimidines via uridine triphosphate. Then you have to go through formation of the deoxyribonucleotides. Then you have to go to DUMP, and then DTMP after methylation. And then there is one enzyme which plays a role, which is absolutely essential as a rheostat, another rheostat to control entry in DNA, which is thymidylate kinase. And thymidylate kinase, this is something very interesting because it is quite often coded in viruses. For example, it's coded in the vaccinia virus. Okay, so this is a story which should be discussed aside, but which may be of interest and linked to what we are discussing now. So you see that C play a role absolutely central for DNA synthesis and non-homothetic growth of DNA. So this has a consequence. Availability is constrained, availability of C is constrained, and this makes that most parasites, intracellular parasites, will progressively enrich in A and T, and they have AT-rich genomes. So this is true. All these stories are about DNA, but I think this may introduce a future story about RNA and RNA viruses. So do we have indications that this is really important? Yes. For example, in the most contracted genome, that of Mycoplasma mycoides SYN 3.0, which is considered as the one of the most compact genome of autonomous organisms, you need to have CTP synthetase. So once again, despite the fact that you do not have the pathways for nucleotide synthesis, this enzyme remains essential. And as you can, if you remember, this was a rheostat in the preceding discussion. So, in summary at this point, you can see that the fact that we use NDPs instead of NTPs alleviates the metabolic pressure on DNA synthesis, but it created a very big problem with C because there is no CDP as a direct product of metabolism. It can only be indirect via degradation of RNA in particular. And you find that there are two rheostats, one which is CTP, I discussed previously, and the second one, which is in thymine synthesis. And at this point, we may need a flywheel because we discussed the importance of flywheels. In fact, in many cells, you have DTDP sugars, which may play the role of flywheels. And again, you find a role specific of thymine here. Why T and not U, for example? And of course, now we have more or less solved the problem for DNA, but what about the membrane? And there is here something which is a puzzle, and curiously, I didn't find any paper suggesting a reason for that. In order to make lipids, you start from CDP containing liponucleotides. That is, in order to make lipids, you need to use C, which is really surprising, in fact, because usually for sugar, for example, you use U or G. So why C for lipids? Okay, and what I will try to show is that this is, again, because C will solve the problem of non-homothetic growth. here is the way of entry of C into RNA. I just alluded to that earlier, but this is a summary of the situation. You start from orotidine monophosphate, make uridine monophosphate, uridine diphosphate, uridine triphosphate, which goes into RNA. But how do you go to C? In order to go to C, you need to have CTP synthase. Okay, and then in order to make DNA, as I showed you, you have to turn over RNA. But CTP is also used for making membranes. This is the bottom of these figures. Again, this also could be coupled to making CMP and therefore using cytosine kinase to go back to DNA. So this means that, especially in eukaryotes, you could use turnover of membrane lipids in order to create proper DNA sources, which is important. But the thing in this picture, you could think that everything is symmetrical and that you could go to cytosine easily. In fact, the thing which is really surprising is that there is at least one enzyme that is every metabolite will age, wear off, and have to be recycled. So when you have nucleotides, in the end, you will produce, for the pyrimidine, you will produce uracil and cytosine. Is it possible to recycle them, to salvage it? For uracil, it's very easy. There is an enzyme which is shown here in blue, which is uracil phosphoribosyl transferase, which makes uridylate directly, okay? And you have the same enzyme for adenine and for guanine, which is hypoxanthine guanine phosphoribosyl transferase. And you would expect to find the same for cytosine, of course. And here is the surprise. I looked into the literature very carefully, and I did not find anywhere a cytosine phosphoribosyl transferase. Okay, there is, you go to Branda, you go to all kinds of databases, you go to papers and so on. There is only one case where some people thought that the enzyme would exist. It is in the amoeba Giardia lamblia. And they said that the enzyme might exist, but the bug has had its genome sequenced. And if you look into the genome, you find that there is no pathway for synthesis of nucleotides, okay? That there is no homologue of phosphoribosyl transferase, which of course is not a complete proof that convergent evolution did not produce the thing, but there is one enzyme which is present there, and it is CTP synthetase again. Despite the fact that there is no way to make nucleotides, you still retain CTP synthetase. And in fact, the people who were working on this bug found that recycling salvage of cytosine went through deamination of cytosine into uracil, and then you go to CTP. So everything points out to cytosine or C derivatives has a very specific metabolites in the cell. They do not, everything is poised as if you need to have a specific control of that. You cannot salvage it directly. Okay, so now let's think about the virus. So here is the complete cycle of the RNA, positive sense RNA virus. So it enters the cell. Because it's a mimic of a messenger RNA, it's immediately translated. The first translation is into an RNA-dependent RNA polymerase, which makes a template, which is copied 50 to 100 times into viral RNA. So it's something very unsymmetrical, but fairly interesting and quite understandable. I won't discuss the different functions of the proteins in the virus, but you can see immediately that there is nothing directly metabolic. So the only thing that the virus can do is tap on the resources provided by the cell. So of course, if there is a problem with C, the virus should see it, in fact. So there are also a few problems, I just mentioned that here, which are linked to the beginning and the end of the virus. Because as you know, when you have to replicate a linear structure, you need to have telomeric-like ends. Otherwise, at each replication cycle, the template would shrink. And so the virus has very structure extremities. In fact, typically, this is the SARS-CoV leader. You can see it's organized in a very specific way. And this is involved in typically replication of the virus and also transcription, because some internal parts of the virus are also transcribed from the negative strand. Okay. Now, when we look into the codon usage, the proteins of the virus and so on, it's fairly complicated. Actually, it's very interesting to study that. We may have to discuss that later on. There is a huge bias in codon usage. And in fact, this is due to the fact that the virus is missing C. That is progressively losing its C content. And this has enormous consequences on its evolution. To my knowledge, people essentially studied the codon usage bias of the virus, but I think we need, and this is type of work that we might like to do, is to make tRNA codon usage bias, because in fact in human cells there are more than 40 different tRNAs to decode the 61 sense codons. So much more than, for example, in mitochondria. So there is a choice of tRNAs which should be understood. So this is the type of work which we need to know to develop, to understand how the virus will evolve. So now, until now, I tried to avoid showing metabolic things. Here is the summary of what I just described to you in terms of metabolism of cytosine with the chemical reactions and the enzyme nomenclature, so you can see how things happen. You will see in this picture that CMP, I don't know whether I can, yes, CMP goes to UMP by deamidation, cytidine to uridine by deamidation, cytosine to uracil by deamidation. This last point, by the way, more or less non-existing in multicellular eukarya, but these two are existing very strongly and have a very important role. So everything is made to be in control of cytosine nucleotide. That is, you cannot have an excess of cytosine. Everything goes back to this enzyme, which is CTP synthetase. So everything is poised in such a way that you control membrane, you control DNA, you control messenger by CTP synthase. And then, here's the thing, CTP synthesis can adjust everything, and it can touch the membrane via CDP-based liponucleotides. And this is also found even upstream of CDP in the so-called CAD multi-enzyme. The pyrimidines are organized in a very complicated enzyme, which controls all the pyrimidines. And this CAD is sensitive to the viruses know that there is a CAD. For example, in antiviral infections, there is a dedicated protein which interacts with CAD and modulates its activity. So this means that viruses, even the very small ones, and enteroviruses are really very small, know that there is something happening in the pyrimidine metabolism. And CTP synthetase is a remarkable enzyme. In fact, it makes filaments. I just alluded to earlier about uridylate kinase, which made filaments in bacteria. But here, this is CTP synthetase. And this so-called cytophilia are located at very precise places in the cells. And they control synthesis of CTP. So CTP is really the handle that can couple DNA synthesis, membrane synthesis, and of course, the cytoplasm. And of course, all this has consequences on viruses. And in fact, what happens, and I will end there that innate immunity already discovered that. A protein which is named viperin for virus inhibitory protein endoplasmic reticulum associated interferon inducible is an analog of CTP, which in fact blocks the synthesis of phospholipids and can also, at least in some cases, use as a chain terminator during replication of the RNA. And so this viperin is induced immediately after entry of a virus in a cell and blocks its metabolism. You can note that, of course, this should be toxic for the cell. And this is a very important thing to remember when we make antivirals later on. They may, of course, be toxic for cells because even in the natural setting, cells make toxic products to control viral infection. You remark that they do not make the non-phosphorylated product, which would be much more dangerous because it could go elsewhere, whereas the 3-phosphate cannot go outside of the cell, or if it goes outside after lysis of the cell, it would not be able to go inside. So it won't contaminate other cells and kill them. There is a second thing. Innate immunity uses a RNA methylase named DNMT2, which in fact normally modifies the anticodon of some tRNAs. I won't discuss that. And at least in the case of viral infection in Drosophila, DNMT2 is used to methylate CpG and block viral replication in Drosophila. And there's a second protein named ZAP, which recognizes some CpG sites in the virus and cleaves the RNA and triggers recruits, in fact, RNases and cleaves the virus. So once again, this is another feature which recognizes C. So as you can see, everything linked to antiviral immunity has been linked to C. So I will stop here, and we have discussed that at the seminar regularly, but we can